Hi, everyone. My name is Will Herrington, not Harrington, that's with an E, like Herrington. So I gave this talk for Paul Stevens, who he and I know each other very well. He's a nephrologist who runs the East Kent Senate, and he asked me to present on this to help people sometimes assimilate evidence. So I said, well, I can do that, but from my particular perspective. And I created 7 rules for them. I think, Mark, you heard this and thought it was interesting, and I'm very pleased to share it a bit more. I'm a professor of clinical trials and epidemiology, but mainly focus on kidney disease. I'm actually a nephrologist, so my practice is kidney medicine. I still do that 1 day a week, but I mainly run large trials. I do have some disclosures. One is I do work for Oxford University, and we do receive grants to run trials from industry, but we don't accept personal honoraria. The other thing is I do work, I guess these are my qualifications for speaking on international and national guideline groups. So I co-chair ESC guidelines and as well as UK Kidney Association guidelines. And I also advise the MHRA from a renal point of view. I'm up here in the top left-hand picture of this map, but I was actually brought up right at the bottom right. So Medway is well known to me. I was brought up just near the creeks in Faversham. So although not a disclosure, it does mean that I know where you all are based in the world. So what I spend my time doing is 2 different types of epidemiology. One is observational, one is randomized. And the observational cohorts, ones that you might recognize very well, these are very passive. I Whitstable, yeah. Some of my cycling routes. The passive observational type studies. Some of them are set up specifically with questionnaires, patients taking samples, and then they're followed longitudinally for disease outcomes of interest. They might be sometimes established as part of routine healthcare data and made available, like CPRD. And these can be enormously large. They're relatively simple to set up, and with routine healthcare data can be easy to follow. They're very helpful for identifying new risk factors, patterns of diseases, and they generally generate hypotheses. But what I'm really interested in and spend most of my time doing is the randomized trials, because they take us to the next step forwards, which is confirmation really of whether or not a risk factor is causal or not. I want to be able to sleep at night when I make guideline recommendations, and I want to be sure that there wasn't a reason why a risk factor was non-causal when we're recommending interventions which have not been tested fully. So I focus most of my energies on interventional studies. So this is interventional studies where we take a treatment or one number of treatments and we allocate them without foreknowledge to patients who could benefit, but we're uncertain about whether or not the treatment is safe and effective. And those types of studies are very intensive to run. We can't do them at the scale of hundreds of thousands, generally speaking. We do them in the thousands, occasionally tens of thousands of patients. And those are the examples of large-scale trials which have really transformed our nephrology practice in the last 15, 20 years, particularly recently in cardiology for years. I don't spend as much time doing observational studies for the reason that they don't give me a definitive answer. I think they're great for describing instances and prevalences of diseases, importance of disease, associations between risk factors. They're quite good in registry forms for identifying post-operative complications. But you'll notice I never really work if you're looking at prescriptions of interventions and how they might associate with outcomes. So I don't do what I would call pharmacoepidemiology, and mainly because I'm interested in interventions with moderate effects. And the problem with observational science is that when you have a moderate effect, we know that these types of analyses are affected by biases, and they're often moderate. Sometimes they're large. Well-conducted studies are often they're moderate. But importantly, we can never exclude residual confounding. And this is supposed to be an amusing slide, but it just gives you an illustration of how, however carefully you manage a database, there can always be reasons why people might differ very substantially from each other, which you can never fully appreciate. So you could take examples of men born in 1948 who are wealthy, a high socioeconomic class, and may have been married twice and have substantial wealth, and then look at them and see that there are things about them which might be quite different in terms of how they might manifest in terms of their health outcomes. Prince Charles and Ozzy Osbourne being extremes end of the spectrum. But this is one of the issues about databases, is that we cannot possibly quantify all of the different types of determinants of our health outcomes to be able to do analyses perfectly and get perfect assessments of effect. So we know that residual confounding is a limitation of all epidemiology, and that's something I have to live with and work around every day. Now, it's not a problem if you're trying to study an intervention which has such large effects that any bias is small. So, for example, people suffering from diabetic ketoacidosis, young children in wards, suddenly insulin arrives on the ward for the very first time, and you see them all slowly waking up with the injections. You don't need a randomized trial to show that insulin is game-changing, just as the same as you don't need it for DC cardioversion, for thyroxine for when they had myxedemic comas, and dialysis as an example, obviously, in our field in nephrology. But most medical interventions are not parachutes like this, which fix a rapidly progressive condition. And so we need our trials really to try to identify treatments with moderate effects. And the reason we, as a trials unit, I work in Oxford, and this has been our philosophy for a very long time, the reason that we think the trials are so important is because we know that if they're sufficiently large enough and well designed, and I'll talk about what we call well designed later, they control the two key errors which limit the ability of observational studies to report reliable findings on interventions. So the first one is, and the most important one, is systematic errors, i.e. biases. But if you make the trials large enough, you can also control play of chance. And both of these types of errors have to be small if you're going to distinguish between no effect and a moderate effect. And that's the challenge most of the time we have with identifying treatments that actually work. And just for those of you that are not used to looking at randomized trials, this is the baseline characteristics of a trial I just reported, which is called the EMPA-KIDNEY trial, which was a drug called empagliflozin versus placebo. And we allocated people with concealment, but with a system of randomization which ensures effective balance for all those key prognostic indicators that we already knew about. So you can see we've got almost exact characteristics between the two groups in terms of age, sex, prior diabetes, and prior cardiovascular disease. But we know by the fact that we've randomized them that anything that's correlated with all of these will also be perfectly balanced. And also we can be pretty confident that every risk factor that we still don't know about, although we can't measure because we've not yet discovered, will also be evenly balanced between the arms. So we establish at baseline this absolute balance between the two groups. So the only difference at the point of randomization is the use of intervention A versus intervention B, or in this case, it's intervention E, empagliflozin, versus a blinded dummy pill. So this is utterly important to us because we know at the beginning of our experiment, we've got perfect control of systematic errors. And as long as we do complete follow-up in all participants long term, we know that we can maintain that balance of randomization, and then we have to make sure we do the appropriate analyses. And I just wanted to show you a few examples now why randomized trials give you a different answer to observational data, so that I can just illustrate to you how robust that randomization process is. And this is an example of statins, LDL lowering therapy, and this is data from Lancet 2020, but included nearly 180,000 people who'd been studied in these trials, enormous amounts of data, often followed for many years. And there was a big question about statins when they came out, about whether or not they were safe. We were doing all-cause mortality studies. People were worried about cancer. And you can see at the end of all of these studies, there's a relative risk of developing cancer within the trial follow-up period of 1.00, so not even the beginnings to show a trend. And importantly, there was 12-year follow-up of some of the really enormous studies of 20,000 patients, and there's no evidence of any effect emerging on cancer. So even if there's a lag, and exposure to statins takes some time before it causes cancer, in actual fact, we don't see any of that developing at all. Yet if you look at the observational studies, and this was published a couple of years later in the New England Journal of Medicine, so one of the most highly high impact medical journal that we report. This is just an observational study looking at real world data from Denmark. The Danish are utterly fantastic at generating routine healthcare data. And they spent some time looking at associations between prescriptions of statins in their community with outcomes. And they reported what I've put in the box here was a suggestion that there may be a reduction in the mortality rate by about 13% for people exposed to a statin versus those not. And this is obviously encouraging if you think that statins are safe, but we've demonstrated that to be the case in the randomized trials already. But what you're seeing here is almost certainly bias, a systematic error of some sort. It's probably a bias by the fact that people who are well were more likely to receive a statin. You can see no dose response. This is on the left-hand side here. The 0.0 to greater than 1.5 is a intensity of statin dose. You could see that the effect doesn't increase as you get exposure to larger doses. So that doesn't sort of fit with a postulate of causality. And then, of course, you can see below here, you've got cardiovascular causes of mortality, which are increased with exposures to statin, which we know is the opposite to what we see in the trials. And again, this is simply what we might call an indication bias or a variety of biases. And this paper stimulated a large amount of editorial because it discussed about observational studies and how they are limited in their ability to assess effects of treatment with small or moderate effects. And in fact, you can see here, we know it's probably a relative risk of 1.00. You can see the size of the biases. They're moderate, 13% effects, but they're still causing erroneous conclusions. And I can go on for days and days about some of these examples. But here's another one which I was interested in at the time I was putting these slides together, which is the effects of atrial fibrillation, warfarin versus placebo. We know that it's highly effective at reducing stroke, and we know that it reduces risk by nearly just over 60%. But if you look at the Danish data, just look at their routine data, look at standard warfarin prescriptions versus those that don't among people with AF, then you underestimate that effect. There's a bias. And then what's even more interesting is if you look at aspirin, it's a much more moderate treatment for reducing risk of stroke. We see 25% risk reductions in the trials compared to, dummy pills. But if you look in the routine healthcare data, then you see a bias, which is actually causing a qualitative bias. You get completely the wrong answer. It suggests that aspirin might increase the risk of stroke, which is implausible. But it just illustrates to you how, when you've got a small or a moderate treatment effect, like a 25% risk reduction, the biases inherent to observational studies actually can cause substantially wrong answers, getting answers completely in the opposite direction. The studies are enormous, so the confidence intervals around that estimate are tight. But that means that it's a precise answer, but it's probably precisely wrong. And so the Danish are fantastic at generating these observational cohorts, and now moving on to realize that they're actually perfect ways to randomize people and then follow them up. So the Danes are now taking their brilliant cohorts, and instead of doing these types of analyses in them, they're now doing amazing trials. And I think there's one at the moment randomizing 300,000 Danes to different types of flu vaccines, and they'll be to very efficiently follow people up. And that would be an appropriate use of these data sources. Whereas I say the pharmacoepidemiological approach, looking at treatments with moderate effects, is not. But the challenge we have as randomized trialists is randomizing enough people. And this is an illustration of some data from, again, the cardiologists, but this time from the clot-busting trials in myocardial infarction, illustrating trials that were done over the 1959 to 1984. So a considerable long period. If you take all of these trials together, then they identified that there was an effect of these clot busters in people with acute myocardial infarction on mortality, about a 22% risk reduction, but these drugs weren't used at all. And that's because if you look at each individual study, there's no clear result from any of them. And so there were designs, in fact, there were two mega trials, and this was one of the ones run by a UK group, the Oxford group, and they tested these clotbuster fibrinolytic streptokinase, but they also randomized people at the same time to aspirin or not. And what they demonstrated that with people who were presenting with acute myocardial infarction, if you got neither, that your risk of mortality was highest at 13%. And it was reduced by about 20% if you got the aspirin, about 20% if you got the fibrinolytic, which is consistent with the previous meta-analysis. But if you got both, you reduced everything by another 30 to further 20%. So you showed that the clotbusters worked. It was a very clear result, which is overwhelmingly positive. And if you look at the use of fibrinolytics after this, they shot up rapidly. But it also demonstrated that we can use them in conjunction with aspirin, and that the effects essentially add. So it's quite rare for us to see 2x2 factorial designs now. People will see trials often study one drug, but this is a really impressive result. And we emphasize the results of that trial importantly to try and make sure that a wide number of people receive fibrinolytics and aspirin. But unfortunately, when people were analyzing the data and it was going through peer review, people started to question the edges of the data, and particularly asking whether or not there were certain types of people who benefited more or less. And in fact, are there some types of people that didn't benefit at all? And there were some subgroup analyses which suggested there were some groups of patients which didn't have a benefit. And these were insisted upon being done post hoc subgroup analyses instead of emphasizing the overall result. And what was interesting, if you look in these papers from the Lancet in 1998 in the subgroup analyses, there's a subgroup analysis in there which you won't see in most papers, which is a subgroup analysis by astrological birth sign. And this was not something that's pre-specified as being scientific subgroup. This was a subgroup which was inserted into the manuscript post hoc because peer reviewers insisted on an additional subgroup analysis. The writers of the paper told me this was a deadly serious subgroup analysis deliberately picked to be something which was not scientifically driven, just to demonstrate how you can pick out subgroups which might appear like the drug is not effective, but in actual fact it may be play of chance. And so if you were Gemini or Libra, the aspirin didn't work, but if you were any other star sign, it did. And that's unlikely to be anything other than the play of chance. I really don't think the star sign you're born under dictates whether or not you're likely to benefit from aspirin or not in the context of acute myocardial infarction. So be really careful when you see subgroup analyses. And this is a subgroup analysis we battled against. An example of a kidney trial we did. This was a statin trial, and you can see that overall the drug was very effective. This was simvastatin versus ezetimibe. It reduced major atherosclerotic events by 17%. You can see we've got these results in males and females, and you can see the results are lined up. I mean, you see no evidence of heterogeneity by sex. But you can see these funny results here where you might start saying, well, it doesn't work if you're young, but it does work when you become 50, and then it doesn't work when you become 60 to 69, then you should start it again when you become 70. Just an example again how random error can make it look like subgroup analyses shift. And the emphasis really must be on the overall result, because no trial is ever powered to look in subgroups specifically. And then to look to see whether or not with these statistical tests for heterogeneity, if there's any important difference across these groups. If there was something important about age modifying risk, then you might see it and you might see a trend. Or you wouldn't expect to see it zigzagging across a group of people ordered like this. So we always encourage people to emphasize the overall result. And we always say, if you're doing meta-analysis, be very careful if people are doing meta-analysis and they're studying subtypes of people from trials. One, it can be data dependent, but secondly, you're ignoring the totality of the evidence. Look at the totality of the evidence first and then start to assess people differently within subgroups. Okay, I'm at rule number 5. I'm going to keep going, Mark, because I know you want me to speak for 20 minutes, but I think I'm nearly there. Because I think there's another illustration from the cardiologists that taught us nephrologists about the importance of waiting for small trials to be confirmed and replicated with large trials. Small trials can be subject to publication bias. They may also not be generalizable because they've often been done in a single center. And this is the example of magnesium in the post-MI period. So there were several trials, 10 trials trying to reduce arrhythmias post-acute MI, which demonstrated there may be reductions in all-cause mortality. And then in 1992, there was a moderately large trial, just over 2,000 patients, which was reported as negative. But in actual fact, if you did a meta-analysis of all of these trials, then you would see a result that was consistent with benefit, and people felt that this was sufficient evidence to start using magnesium. But there was a very large trial in the offing, unbelievably large trial of 58,000 people called ISIS-4, which had been assessing magnesium versus control. And when it reported, it found absolutely no effect of magnesium versus no magnesium in patients who presented with acute myocardial infarction. People had been so convinced by the magnesium small trial arguments before, there was still some doubt about whether or not this result was the truth. There was a concern that it might have been certain low-risk patients had been involved in the ISIS-4, and therefore it biased the result to the null because they had lower event rates, less likely to have a benefit. Or it might also be that the magnesium was infused later after the acute myocardial infarction. And so a further trial called MAGIC was designed, which was designed to identify those with ST elevation MI, so at the highest risk, so you'll see the event rate is high, but also have prompt infusion of magnesium. And again, they got a completely null result. And if you meta-analyze all of the trials, you see there's no effect of magnesium. So be very careful of the first results coming out. Even if over a series of small trials you see benefit, we really must do large trials across multi-centers to essentially validate a result. Rule number 6, we're almost there, Mark, is blinding. So in some cases, blinding is not needed because you might report, for example, deaths, and you might have a perfect way to ascertain death in everyone, like in the recovery trial of COVID-19 in the UK, 30-day mortality. Everyone's death is reported. We can link to registries. We know we can ascertain death. And if the outcome is just all causes of death, then you're not going to get any reporting bias. But where the outcome is really important and it is subject to observer bias or some other behavior could affect it, is it really important to blind. And here's a really nice example. And I'm going to flick through 3 slides very quickly just to illustrate the point, but I encourage you to read the Simplicity HTN studies, because these are renal denervation studies, and renal denervation was starting to be used and is still used in some centres. But essentially, it started off with observational studies like our one I described at the beginning, simply observing people who had renal denervation and what happened to their blood pressure afterwards. So they were selected because they had high blood pressure, and then they had their renal denervation, and then their blood pressure was observed afterwards. Now, if you select anyone with a high blood pressure and just observe them, on average, their blood pressure will fall down. It's a statistical phenomenon called a regression to the mean. But this effect in Simplicity HTN-1 was so large, a 30 millimeter reduction in systolic blood pressure, people assumed it must be due to the renal denervation that all these patients had. In actual fact, when they went on and did a randomized trial and randomly allocated renal denervation, so there's a zapping of the renal arteries to try and remove the nerves, and it's thought to reduce blood pressure. By doing this in 106 patients, some who had no denervation versus those that did, they actually saw similar reductions of 32 millimeters of mercury in systolic blood pressure for the primary endpoint. So this looked like it was a really effective treatment. Brilliant if you want to reduce the number of pills, a single zap, a bit like a vaccine for blood pressure, and you can treat people. But the definitive, I think probably the most definitive answer to this, although more studies are ongoing, was a blinded trial. So the Simplicity HTN-2 study was open label, so people knew that they'd been allocated to renal denervation or not, and so did the people measuring their blood pressure. Whereas if you could do sham renal denervation, and everyone in this trial, Simplicity HTN-3, went into the radiology department, had their renal arteries cannulated, only some of them had the denervation process, others did not. They were wearing headphones. The only person that knew whether or not the electrodes were connected was the person performing the procedure. And then all the follow-up was done externally by research coordinators and patients who were completely unaware of their treatment allocation. When you do this, they found absolutely no significant difference in the blood pressure between those that were denervated versus those that went through the sham procedure. So people have called this as regression to the truth. There must have been some systematic bias in the way the blood pressure was being ascertained, or the behavior of patients just before their clinic appointments, that led to the Simplicity HTN-2 result, a randomized study which overestimated a treatment benefit. So beware at open label studies, because they can introduce bias even when you think it is something which is measured perfectly. And then the last point, I think I've got 1 minute, I can just say this, is that something which I am increasingly aware of, and we should be increasingly aware of, with the changes in the world at the moment, but that is the impact of commercialism into research. I think it's a major positive thing, because we're able to do in nephrology now the size of trials we needed to be doing many years ago in a relatively rare group of patients, and we can generate really reliable evidence. And I do trials. I propose trials to industry and I work with them closely, but we're very cautious about when we see an industry funded trial to make sure that they've designed a trial which is widely generalizable, so it's got the public health at its primary objective. You'll see too many trials nowadays with very long lists of exclusions, which make the external generalizability of the trial reduced. If you select out patients who are low risk of a safety outcome, you're going to essentially generate a result more likely to show benefit and less likely to show harm, and we will be left with difficult decisions when it comes to risk benefit balances. But also, commercial research can be incredibly expensive. They're so concerned about audit findings and inspection findings that there is complicated operations in the trials, which can actually lead to very expensive development costs. And the contract research organizations now are highly profitable, with revenue growing each year. And this money obviously leaves research because it's not invested directly into the hospitals. It's into the people who are running trials within corporate organizations. And then the last thing is that obviously industry want to try and generate income from their interventions as early as possible, and so they try and work out within the regulations what is possible. A very large, simple trial following people for the long term will generate the best safety data and the best information for us as clinicians, but it will be a long lag in terms of a company, in terms of generating revenue back. And so the orphan drug designation, studying a drug in a rare disease, has become quite common, even if the drug might benefit large numbers of people. And there's very interesting data from the pharmaceutical industry looking at the revenue of the top selling drugs in the US. And it's very clear that over the last few years that revenue has increased, but the types of drugs which are in the top 10 sellers have changed very dramatically, from those types of drugs which might be treating the population at large at risk, down to those which might be treating much smaller proportions of people. And the numbers of people who are eligible for the use of the drugs is estimated to be about 7 to 8 times less. So of course the wider public benefit is lost when that type of priority is considered. So as a nephrologist, I'm interested in rare diseases, but I'm also interested in the public good, and there's a real balance to be struck. So these are things to consider when you look at research. I'm just going to leave these up on the screen. These are the 7 rules I presented as we've gone along, and I'll pause now for any questions, Mark.